lecture, wherein in this video, the discussion will focus on protein synthesis, particularly on transcription. So without further ado, let's get started. Transcription is the process by which DNA is copied to mRNA, which carries the information needed for protein synthesis. Transcription is the first step in gene expression in which information from a gene is used to construct a functional product such as a protein. The goal of transcription is to make our RNA a copy of a gene's DNA sequence. For a protein-coding gene, the RNA copy or transcript carries the information needed to build polypeptide or protein subunit. The main enzyme involved in transcription is RNA polymerase, which is a single-stranded DNA template to synthesize a complementary strand for RNA. Specifically, RNA polymerase builds an RNA strand in the 5-3 direction, adding each new nucleotide to the 3 end of the strand. Transcription takes place in two broad steps. First, pre-messenger RNA is formed with the involvement of RNA polymerase enzymes. The process relies on Watson-Crick base pairing, and the resultant single strand of RNA is the reverse complement of the original DNA sequence. The pre-messenger RNA is then edited to produce the desired mRNA molecule in a process called RNA splicing. Formation of pre-messenger RNA. The mechanism of transcription parallel in that of DNA replication. As with DNA replication, partial unwinding of the double helix must occur before transcription can take place, and it is the RNA polymerase enzymes that catalyze this process. Unlike DNA replication, in which both strands are copied, only one strand is transcribed. The strand that contains the gene is called the sense strand, while complementary strand is the antisense strand. The mRNA produced in transcription is a copy of the sense strand, but it is the antisense strand that is transcribed. Ribonucleoside triphosphate or NTPs align along the antisense DNA strand with Watson-Crick base pairing, where A pairs with U. RNA polymerase joins the ribonucleotides together to form a pre-messenger RNA molecule that is complementary to a region of the antisense DNA strand. Transcription ends when the RNA polymerase enzyme reaches a triplet of bases that is read as a stop signal. The DNA molecule rewinds to reform the double helix. The pre-messenger RNA is trapped to remove the introns and create messenger RNA in a process called RNA splicing. The pre-messenger RNA thus formed contains introns which are not required for protein synthesis. In alternative splicing, individual exons are either spliced or included, giving rise to several different possible mRNA products. Each mRNA product codes for a different protein isoform. These protein isoforms differ in their peptide sequence and therefore their biological activity. It is estimated that up to 60% of human gene products undergo alternative splicing. Alternative splicing contributes to protein diversity. A single gene transcript RNA can have thousands of different splicing patterns and will therefore code for thousands of different proteins. A diverse proteome is generated from a relatively limited genome. Splicing is important genetic regulation alteration of the splicing pattern in response to cellular condition changes protein expression. Perhaps, not surprisingly, abnormal splicing patterns can lead to disease states including cancer. In reverse transcription, RNA is reverse transcribed into DNA. This process, catalyzed by reverse transcriptase enzymes, allows viruses including the human immunodeficiency virus to use RNA as their genetic material. Reverse transcriptase enzymes have also found applications in biotechnology, allowing scientists to convert RNA to DNA for techniques such as PCR. The stage 1 in transcription is initiation. Transcription is catalyzed by the enzyme RNA polymerase. It attaches to and moves along the DNA molecule until it reaches a promoter sequence, which indicates the starting point of transcription. There may be multiple promoter sequences in a DNA molecule. Transcription factors are proteins that control the rate of transcription. They too bind to the promoter sequences with RNA polymerase. Once bound to the promoter sequence, RNA polymerase unwinds a portion of a DNA double helix, exposing the bases on each of the two DNA strands. The stage 2 for transcription process is the elongation. One DNA strand, which is the template strand, is read in a 3 to 5 direction and so provides the template for the new mRNA molecule. The other DNA strand is referred to as the coding strand. This is because the base sequence of the new mRNA is identical to it, except for the replacement of thiamine bases with uracil. Incoming ribonucleotides are used by RNA polymerase to form the mRNA strand. It does this using complementary base pairing. Incoming ribonucleotides are used by RNA polymerase to form the mRNA strand. It does this using complementary base pairing wherein adenine is paired to uracil, thymine is paired to adenine, cytosine is paired to guanine, and guanine is paired to cytosine. RNA polymerase then catalyzes the formation of phosphodiester bands between adjacent ribonucleotides. Bases can only be added to the 3' end, so the strand elongates in a 5 to 3 direction. The stage 3 of transcription is termination. In this stage, elongation will continue until the RNA polymerase encounters a stop sequence. At this point, transcription stops and the RNA polymerase releases the DNA template. And that's it for the discussion of protein synthesis, specifically on the process of transcription. I hope you learned something from this video and have a good day.